Hi everyone, welcome back. I hope you enjoyed the first section of the class. And we're going to start this series of lectures that contribute to the second midterm today, chapters 7 and 9, 10, and 11. We're going to talk about the sensory motor systems of the brain and also about the general research methods today. Then we'll have our second midterm before, after, and during reading week. So today we're going to talk about how scientists and labs at the university and around the world study the brain through the study of its structure and its function and their link to behavior. And we'll talk about a number of different technologies today, including electrical recording of the brain, anatomical imaging of the brain, functional imaging of the brain, and then some other chemical and genetic measurements that we can make. Compare them all together, and we'll talk briefly about using animals in research. So neuropsychology is a study of a relation between brain function and behavior. And neuropsychology as a field generally has an emphasis on humans and dates back hundreds of years to the times of Paul Broca, when they were finding patients with particular cognitive deficits, waiting, studying them and waiting until they died, and then taking an autopsy and looking at where they had damage in their brains. And this led them to understand that the left frontal lobe region is related to language. And the way that we can measure links between brain and behavior includes such things as non-invasive imaging like fMRI, neuroanatomical measurements like estimating people's brain size after they're passing away, and also studying their behavior. One of the first techniques used to study the brain was histology, as we've already talked about, which is the slicing of tissue into very thin slices, and then the preparation on slides and the study through different types of microscopes. Different types of stains and dyes and microscopes accentuate different parts of the anatomy. And new techniques, contemporary techniques, also can image the neurochemical and molecular changes in the cell, as well as microscopic morphological changes, and relate these to behavior. So here's 4 different imaging technologies. On the left, a light microscope, something that you could prepare in any given science lab. You could stain brain tissue with particular type of stain to accentuate the cell bodies like this, and then put it on a slide and look at it under a microscope. Alternatively, here's a light microscope with a very high magnification, allowing us to look at individual cells based off a different stain, such that we can even look at the arborization of their dendrites. An electron microscope, something we talked about in the first midterm, allows imaging of objects that are smaller than the wavelength of light. Here, imaging the vesicles in the synapse, in the synaptic cleft, in the presynaptic and postsynaptic neurons on either side of the synaptic cleft. And some of the newest technologies are multiphoton microscopes, in which they can image cells even in live, awake and behaving animals. So some simple ways to think about the link between brain and behavior is by studying just the link between this anatomy, cellular anatomy, and behavior. So we can think about how animals trained on different types of learning tasks might be influenced by the organization or the amount of different cells in particular parts of their brain. So learning, we know, can lead to modifications of synaptic organization of cells in particular cortical regions, and the number of newly generated cells that survive in the hippocampus can influence our ability to learn. The textbook gives an example study showing a normal, healthy rat hippocampus under a microscope image showing in white all the healthy cells here in an area called the dentate gyrus. And in an animal model of degeneration of these new neurons, in which they give a particular drug that decreases the production of new cells in the dentate gyrus. Under a microscope, they observe that the manipulation worked and there's less cells in this area. Then they can give these a different animal that has the same manipulation who's still alive. They can give them a learning task and see how that difference in the number of cells in the hippocampus that they've induced influences their learning. So here's a particular type of memory task that has been given to rats, in which they're given two sets of objects to remember in two different contexts. So stars in a square room and hexagons in a circular room. The rat studies this for a number of days, and then they're given a test phase in which both, as for instance, in a square room, both the normal object and a new object are present, and in a circular room, vice versa. And so the dependent measure is how long the mouse explores each of these two objects. Are they going to explore the normal object that has the right context for that room, or the novel object that wasn't studied in that room? And normal rats study the novel object more, and they spend more time investigating the novel object. These rats with cell loss in their hippocampus performed at chance. That is, they looked at each of these objects just as much, irrespective of whether they were in the right context. So that's a great example of a very simple manipulation that can be done to study the relationship between the brain and behavior. Behavioral neuroscience is another related field related to neuropsychology. It's the study of the biological basis of behavior and is done in both humans and laboratory animals. It's difficult to create tasks that allow us to study both normal and abnormal behavior. So development of new methods that allow manipulation of behavior in nuanced ways is important. Neuropsychological testing is often done as a technique in cognitive neuroscience or in neuroscience. So for instance, people with damage to their temporal lobes present to the doctor with memory disturbance, that difficulty in certain types of memory. However, different types of memory are influenced by different types of damage to the brain, and not all memory is the same. So memory for events, colors, names, and different types of motor skills are all different. And we'll learn more about this in our chapters on learning and memory later in the course. So we have to measure each type of memory separately in certain types of tasks. So here's three examples of different tests for human memory that measure different types of memory. Here's one that measures short-term recall of spatial position. So can you remember where things were organized over a short period of time? The examiner taps a certain sequence of these numbered blocks, and the patient or the research participant has to follow that same pattern, but without seeing the numbers, just from the spatial layout alone. And the researcher can identify how many of these blocks the patient or participant can remember. Another type of memory task that's used to measure a different type of memory is called a mirror drawing task, in which one learns to trace an object through the reflection of a mirror. So they don't see their own hand, they only see the reflection of their hand in the mirror. So they have to learn to adjust their motor output depending on this flipped feedback. And slowly over time, participants get better and better at this. They make fewer and fewer crossing errors. And we call this a procedural memory, much like riding a bike or learning to play piano. Some patients that have no recollection of having learned this task because they have damage to their hippocampus, nonetheless retain an ability to get better and better at it. A really good example of these two different memory systems in the brain at play. Here's another example of a human memory task in which participants are given a series of playing cards with objects on them. When a question mark appears, the participant is asked which one of these the objects they saw most recently, and they're forced to make a choice between the two. And this can be used to measure how recently in time people can recall objects. But also it's been shown to, for example, show that even in situations where people are completely guessing, where people say they have not familiar at all and have no idea which of the objects they've seen before, they're able to guess above chance, indicating that we don't have a full level of conscious awareness of all the abilities of our memory system. Again, showing these multiple different memory systems coexisting in our brain. Another set of behavioral studies has been done on rats using something called a Morris water maze, a large pool of milky water that rats can swim around in and try to find platforms. This allows for nuanced study of different types of memory systems by manipulating the brain of the rat before or after they take part in these tasks. So for instance, in a place, the rat has to learn where this platform is in the milk. So in place learning task, the most simple task, the rat uses the cues around the room to learn where the platform is. We'll see some pictures on the next slide. In a matching to place learning task, on every given day, this platform changes, but then it continues to be in the same place throughout the day, allowing researchers to study not only how fast the rat can learn how to find the platform on any given one day, but how they can improve at this general strategy of learning to find the platform over multiple days, even if it changes in location. And finally, researchers have used a landmark version of the task in which the platform is only identified in its location by landmarks on the distant walls of the room or the local side of the tank. I'll show you some pictures on the next slide. So here's an example of a little rat swimming in this milky water. The rat's motivated to get out of the water and find the platform. The rat uses room cues around the room to identify the platform and repeatedly swim there, such that on the fourth and fifth trial, it gets faster and faster to find the platform. If you replace the platform on each day, then of course rats take some time to find on each day. However, they get better and better at this task over time. Then you can manipulate their brain and identify which of these two types of learning are influenced. Finally, researchers can teach rats to ignore the general room cues and identify just local cues on the wall of the tank. So they might spin the tank around or put a cue on the tank in different places and put the platform nearby. This is a different type of learning that involves different circuits in our brain. Much like if I tell you to go downtown near the legislature building, this is a landmark learning task, as opposed to you learning randomly where it is over time. Another technique that researchers can use is to break down certain movements that we make into a series of discrete units so that we can identify which of those units might be damaged by certain brain perturbations or might be improved by training. So the authors of the textbook have done a study in which they study the skilled reaching of rats through a narrow cavity in their apparatus. And so they have to reach through and grab food and then pull it out and turn it just the right way. And they can break these movements into different parts. So they can see the rat aiming its paw, reaching for the food, grasping the food, and withdrawing and moving it to the mouth. They can use video to distinguish these parts and identify which of these parts of the movement might be influenced by certain brain damage or manipulations. This also shows how dexterous even a small animal like a rat can be if we zoom in space and time. The textbook even shows this new technology of rat touch screens that can be used to create learning tasks for rats that can be very similar to those used in human research. So in addition to studying these behavioral outcomes, we want to sometimes manipulate brain activity in order to make causal claims about what those brain areas did for those behaviors. So we can modify the brain and then measure what the outcome and behavior is. This will allow us to develop hypotheses about how the brain influences behavior and test them. And we can also develop animal models of certain disorders or neurological problems that we can then use to develop treatments or drugs. So, for instance, Parkinson's patients, we've learned, have this motor disturbance that's associated with cell death in the substantia nigra of the midbrain of their brainstem, here shown in orange. Parkinson's patient has a degradation of these neurons such that if you look in a microscope after they pass away, they'll have a much lower cell count. So researchers can experimentally injure these regions in laboratory animals and then study what the outcomes are on behavior and study possible treatments. Lesioning the brain is a very long, popular technique of studying the relationship between brain and behavior. Carl Lashley, 100 years ago, was trying to identify the location of memory in the brain, and he slowly cut different sections of the brain, but without cutting the hippocampus because of its being tucked away under the cortex, he didn't find any particular area that influenced memory. It wasn't until a couple decades later when a surgeon named Scoville removed the hippocampus from a patient named HM to treat their epilepsy. And when this hippocampus was removed, HM lost their ability to produce new memories, a disorder called amnesia, showing the important role of the hippocampus in this cognitive process. And the substantia nigra has also been lesioned as a way to study Parkinson's disease, as we saw in the previous slide. We can make our brain lesions more precise in animals and also in humans, for an example of overactive tissue, by using stereotaxic apparatus. So here are two examples in animals and humans where we can identify with an MRI image or with an atlas a particular 3D location where we know that we need to create a lesion in the brain, and then we can directly target that area and create that lesion. Researchers can also use electrodes to lesion the brain. They can lower the electrodes in and pass current through at a sufficient voltage and amplitude that they can destroy the tissue in the region. Now, after these lesions are induced, and even in human brain injury, compensation occurs. So our brain is plastic and it modifies behavior from that used in the past, and it modifies which circuits it's being used to produce certain behavior. So to avoid this compensation and to allow animals to recover fully from the experimental technique, we can induce temporary and reversible lesions instead, where we could, for instance, regionally cool a particular part of the brain or administer a GABA agonist and increase inhibition in a particular section of the brain, and then study how that influences behavior. In addition to lesions, we can also electrically or using other techniques stimulate the brain and increase the activity of certain areas. This was first used by the Canadian neurosurgeon Wilder Penfield in Montreal to stimulate the cerebral cortex of humans during neurosurgery. He wanted to understand what the function was of the particular areas he was considering cutting out of their brain, and so he woke them up during surgery and stimulated their brain while asking them what they felt. We can do this in animals. For instance, rats with electrodes in their hypothalamus will heat whenever you stimulate that electrode, so you can induce the circuits that and drive the animal to eat. And you can also find certain areas in the rat's brain where you can put an electrode that when they press a lever activates that electrode, and you can find rats that will continuously press that lever to self-stimulate, indicating that pressing that electrode seems to be rewarding by stimulating that area of the brain as a way to study addiction. You can stimulate the adjacent area after a stroke, and you can see improvement in motor behaviors. And another example is if you electrically stimulate specific brain areas of a rat in a Parkinson model, you can restore some motor deficits. In humans, two different types of electrical stimulation are commonly used. Deep brain stimulation is now a very common technique in which an electrode is implanted in the brain, much like a pacemaker, and a low voltage electrical current is applied with particular rhythmicities to facilitate particular behavior. This is common in Parkinson's and in depression and OCD treatment for very difficult cases. Transcranial magnetic stimulation is a much less invasive technique in which a magnetic coil is placed over the skull of an awake human, and it creates this large magnetic field that disrupts the electrical activity in the area underneath. This can induce increase or decrease in the activity of circuits there. So this magnetic field can pass through the skull, and it causes the population of neurons underneath to fire more and by depolarizing them. It's been shown to be useful for treatment of depression because, as it can activate certain frontal areas with repeated stimulation. And there's two techniques. You can use a pulse of stimulation, or you can use repetitive stimulation called repetitive TMS. We can apply certain drugs to humans or animals and then measure what happens in their brain and behavior. So by administering a drug that passes into the brain, we can also put a cannula embedded into the brain so that the drug doesn't have to pass the blood-brain barrier and can directly target particular brain areas. Drugs like local cooling or GABA agonists have a beneficial effect in that they wear off, and that we can bring the animal back to baseline and study how the function changes. Some new technologies to stimulate and manipulate brain activity include optogenetics. Optogenetics is a technique in which certain receptors are embedded onto the cells of animals experimentally, and those receptors, when light is shined on them, allow ions to pass through, depolarizing or hyperpolarizing the cell. So optogenetics is a transgenic technique. So it combines genetics in order to make those receptors expressed on the particular cells using the virus, and it uses light to target those receptors. And it's based on the discovery that light can activate certain proteins, these light receptors. And these proteins can occur naturally, or usually they're experimentally inserted into cells, and then a fiber optic can be delivered into a particular brain area such that only certain brain areas get shone with light, and such only those cells get activated in this optogenetic technique. Here's two examples of the types of receptors that can be added into viruses and made to be expressed in the cells of animals in particular brain areas. Channarhodopsin, CHR2, when exposed to blue light, it opens up an ion channel and it depolarizes the cell, making it more excited and making it fire more action potentials. Alternatively, this other type of receptor, when green-yellow light is shown on it, it creates a chloride pump that hyperpolarizes the cell and makes it less likely to fire action potentials. So you can imagine the experimental control possible by manipulating these amounts of these receptors in certain types of cells. In fact, you can put the receptors in specific neuron types by targeting the genes of those types. So for instance, you could only have these receptors in pyramidal cells, such that when you shine light on an area, only those pyramidal cells will be activated. So this is really a useful technique because it has a high resolution in space, because you can target particular brain areas with the fiber optics, and with time because you can turn on and off the light whenever you want to control the cells. Some even more advanced technologies involve creating custom receptors that you embed into certain cells that then have a paired up custom drug that only activates those receptors, or they only get activated by that drug. This is called designer receptor exclusively activated by designer drugs DREAD technique. So it's a G-coupled protein receptor, and it responds only to these drugs and then creates this cascade of cellular processes when the drug is present in the system. Okay, next we're going to go through some of the techniques for measuring the human brain in more detail. This is what I focus on in my research here at the University of Alberta, so it's of interest to me. We measure the brain's it's of particular interest to me. We measure the brain's electrical activity in humans in a number of ways. And we'll start talking about single cell recording, which we've already mentioned in animals, and then zoom out to the human and think about how we can measure their brain activity from big groups of their neurons firing together. So we'll talk about electroencephalography, EEG, event-related potentials, a way to analyze EEG data, and magnetoencephalography, a different technique that uses magnetic fields. So we can record action potentials from single cells by placing fine electrodes, microscopic electrodes, down into the inside and outside of cells, or into the extracellular space around cells. And we can use these techniques to even distinguish as many as 40 neurons in a local area, all firing, and see when different ones fire at different times. So we can measure intracellularly to isolate a single neuron and study when it fires and when it doesn't as well, and how that relates to its ongoing resting membrane potential. And some of the most famous research that has come out of the cellular recording won the researchers the Nobel Prize for their identification of these cells in the hippocampus called place cells. Place cells are cells in the rat and mouse and purportedly the human hippocampus that fire when an animal is in a particular part of their environment. Like if you were in the university, you'd have some cells that fire whenever you get to sub, and a different cell that fires whenever you get to hub, if we have analogous cells in humans. And these cells allow for the coding of the spatial location of the animal and allow us to create a spatial map of our world. Different cells have been identified in addition to these place cells, such as head direction cells that fire whenever we face a particular direction in the world, and grid cells, which fire in these repetitive grids that seem to allow for measurement of distance moved. Electroencephalography, or EEG, is the measurement of the electrical activity from the scalp or the skin on the head of humans and animals. It measures generally the graded potentials, the summation of graded potentials, that is the IPSPs and EPSPs in the dendritic trees, not necessarily the action potentials. And it measures these sum graded potentials from thousands of neurons simultaneously, much like you were measuring the outcome of a hockey game by just sticking a microphone on the roof of the Rogers Center. So by doing this, even though we might not learn much about the game or the brain in this case, we can still identify changes in electrical activity that seem to relate to behavior more generally. So as our behavior changes, these broad measures of electrical activity from the outside of our head change. And they often display patterns. Some of them are rhythmic, that is, they go up and down like waves. You've heard of these brain waves before. And when someone's alive, their brain activity is never flat. There always is this churning of electrical activity in their brain, even if they're in deep sleep or comatose. So here's an example of electrodes attached to the outside of the scalp of an individual, and the electrical voltage between two of these electrodes is then measured. And just like the voltage between two sides of your battery is 1.5 volts, these small microvolt measurements from the head oscillate or go up and down in time rhythmically over time, like as if the battery's voltage was changing. And these are created by large groups of neurons giving off electrical fields all synchronized together. So I think I've already covered this slide. It's on here again. So we can see if we look at the EEG in different states of an individual that it changes. When they're awake, it's low amplitude and fast in frequency. As they become drowsy, it slows in frequency and increases in amplitude. When we're relaxed and our eyes are closed or dazing off, we create these things called alpha waves, which I've studied a lot in my research. These large 10 times a second oscillations in the brain that seem to be related to inhibiting our incoming sensory information. As we fall asleep, we get these slower and higher amplitude waves. In deep sleep, we get very slow coordinated waves across the whole brain, which are even more accentuated in someone that might be in a coma. So EEG we can see as an indicator of not just arousal and relaxation, these general changes throughout the day, but can also be used, for instance, to measure our sleep stages. It can be used to measure how much anesthesia we need during a surgery. It can be used to estimate the severity of a head injury and to identify brain abnormalities like epilepsy. So it's a very useful tool for research, what we do in my lab, and also for diagnosing dysfunction of the brain. Just yeah, there we go. Okay. Just wanted to show that other technology. Wanted to make sure it was here. So as we measure this ongoing electrical activity, when things happen, like we hear a sound or a flash of light happens or someone calls our name, the electrical circuit that represents that stimulus fires and there's an electrical field created, but that electrical field is often much too small to be seen in the large ongoing electrical activity of the brain. However, if we use repeated stimulation, so we create the same stimulus over and over again and we present it to someone, we can average the electrical activity during those repeated stimulations to create a picture of what the brain's response is without all that noise clouding it. These are largely, again, created by the graded potentials on dendrites when we have incoming sensory stimulation. And they all start at the time of this incoming sensory information. So they're event related. And as I said, they're averaged together. So here's an example of a single EEG recording from the onset of a sound. 10 recordings averaged together, 50 and 100. So when we get to 100 recordings, we can see now that the electrical activity caused by certain regions of the brain as the sound is processed is observable in this measurement from the outside of the head. And we can then identify the time that it takes certain processes to occur and the amplitude of those processes as estimates of those cognitive activities. So you can not only study, therefore, by identifying which regions of the brain you see these responses being the largest, you can identify roughly areas in which there's increased or decreased activity in response to stimulation. Then you can create these colorful maps of brain activity of where there's maximal activity. But you can also use EEG. What it's most beneficial for is to identify the temporal stages of during which certain processes occur. Because of its great resolution in time, it can measure things at a very fast resolution. So you can identify which area, which process occurs first and which process occurs second. And this technology is becoming more and more miniaturized to the point where now you can buy something for a couple $100, like I have here, the Interaxon Muse, which I use in one of my other classes in a lab where the whole class gets to learn about EEG and ERPs. That's a 375 class that I've taught sometimes. So portable EEG can now be used both by the consumer as a way to help us relax our brain and meditate, but also we can use this new technology as a way to study the brain outside the lab. So a lot of my research, we take these really portable EEG devices and put them on people in new situations like riding a bike, or we've taken it to the hospital and measure in the hospital bed from people very acutely, and we're now putting it in the ambulance. When the ambulance runs out to a suspected stroke, we can put this EEG on them and measure their electrical activity of their brain. Related to EEG is a technology called Magnetoencephalography, or MEG. When the electrical activity of a neuron occurs, you might recall from physics that the right-hand rule creates a perpendicular magnetic field. So if this is the electrical field, then a magnetic field occurs orthogonal to that electrical field. And that magnetic field can also be used, or that magnetic field generated by the neurons perpendicular to the electrical field can be measured by precise magnetosensors placed on the outside of the head. So we place a bunch of these detectors on the outside of the skull, and we allow for a 3D localization of electrical activity that's more effective than with EEG, because of the magnetic activity is less smeared by the scalp and skull. It, however, does have a high cost, and importantly, it is not very portable, but newer technologies are allowing for recent advances in this tech. Next, we'll talk about anatomical imaging. So can we create pictures of the anatomy of individuals and use that to study the relationship between their brain and behavior? Some of the earliest technologies that were used to identify the anatomy of the brain were CT scans, which are X-rays that are combined together using computer technology from multiple angles. So an X-ray beam is passed at many different angles through the brain, much like you might walk around a mountain to try to understand its shape, and in 3 dimensions. And these images are then combined to create 3-dimensional images of the brain, which, for instance, can be used to identify lesions. Still today at the hospital, many CT scans are ordered. So the dense skull forms a white border on the CT scan, and the gray and white matter doesn't show much differential image. So you can't distinguish the two from a CT scan. And the ventricles can be seen, and this can be therefore used to identify any abnormalities in the ventricles or cerebrospinal fluid. And some of the major fissures are rendered a little dark, so you can identify some of the anatomy. However, a newer technology was developed in the 1980s called magnetic resonance imaging. Magnetic resonance imaging allowed for a much more precise representation of the human body by producing these 3-dimensional images, slices through the tissue, which can be recreated into a 3-dimensional image. The MRI is produced by placing the human in a very strong magnetic field that aligns all of the protons in their brain tissue, and then a radio wave that misaligns those protons briefly, and then also measures the electrical activity that they give off as they snap back into alignment. And depending on the substance in the tissue, there'll be different amounts of these protons. And so the time it takes them to snap back into alignment will be slightly different, for instance, for white matter or gray matter or cerebral spinal fluid or bone, And so these times can then be used to recreate an image in 3 dimensions of the tissue, which is where we see these MRI images. So if you've ever been in an MRI for a knee or a spine injury, you know that you go inside this large magnetic coil, and there's a huge magnetic field running this way through the coil along the bore, aligning all your protons together, and then another coil wrapped around your head or neck or knee that stimulates those protons out of alignment and then measures the time it takes for them to get back into alignment. Here's a recent MRI I just had of my spine at the hospital in order to identify the source of some long-term pain I've been having. And I'll talk to you more about this in a couple of weeks in the chapter on somatosensation. We'll go over the parts of the spine, and here's the spinal cord, the very bottom of the spinal cord, and these long axons coming down the spinal cord, sending motor signals from my brain into, then they would exit here and go out to my body to move my muscles. So magnetic resonance imaging, as we've mentioned, measures these electrical currents created by the wobbling atoms, and different types of atoms give off these currents differently. Different types of tissue have different amounts of protons, and so create different electrical signals when the wobble happens. Here's a cartoon that's not from the textbook that might help you visualize this a little bit. So any one of us right now has just randomly oriented spinning protons in the MRI. They all get aligned, and some of them one way, some the opposite way. An electrical field can be added to shift some of those into the opposite direction, which is a state of high potential energy, which then that energy gets given off as they snap back into their original alignment, and that energy being given off is what's measured by the coil and turned into the image. MRI can be used further to estimate the white matter tracts in the brain by using a technique called diffusion tensor imaging, or DTI. DTI estimates white matter tracts by looking for areas in which water molecules are moving in one direction because they're stuck inside an axon and can't go in the other directions. And by identifying the direction that water molecules generally diffuse in each part of the brain, they can recreate maps of where the axons were headed in those different areas based off the direction of those diffusions. This can be used to delineate abnormalities in white matter pathways, for instance. Finally, magnetic resonance spectroscopy is using an MRI machine to identify the chemical properties of the brain, not just the anatomical differences, but for instance, how much dopamine or how much glutamate or GABA might be in a particular brain area. This is useful following concussions or brain injuries in which the metabolites built up seem to be related to the outcome. Next, we're going to talk about functional brain imaging. So can we use images of the brain to study brain activity over time, usually using blood flow? So when a brain is active, in addition to giving off this electrical signal we can measure with the EEG and the MEG, the amount of blood, oxygen, and glucose coming into that area increases. This is signaled by certain astrocytic processes. And because of this increase in blood, we can measure brain activity's echo by measuring where blood flows after it happens. So if we find a particular area where there was more blood flow in a particular task, we know that the neurons there were more active and therefore recruited more blood to get the oxygen, glucose, and iron that they need. So functional magnetic resonance imaging is the study of the changes in human brain's blood flow. And it's afforded by the fact that when our brain requires new blood, it actually gets more than it needs. And this increase in oxygen in the tissue, this overcompensation, can therefore be measured by changes in the magnetic properties of those protons as they snap back. So differences in the MRI signal can be used to identify the amount of blood in an area. And the oxygenated blood coming from our lungs has different magnetic properties than the deoxygenated blood that our body has already used. So we can even estimate the oxygenation of the blood to identify areas in which our brain was most active. So in the most simple example of a task like this, one can stimulate, for instance, with light. So one can show an individual a flashing light when they want and then turn it to complete darkness. When the light is off, you can measure the magnetic properties of the brain. And then when the light is on, we can continue to measure and we can see that the MRI signal intensity, that is like how, because there's now blood flowing to that region, there's more protons and a larger signal given off when they all snap back in the magnetic field, that MRI signal intensity increases, but it's localized to the regions that process that visual stimulus. It's not in the auditory areas or the motor areas, it's in the visual areas. When the light turns off, it goes back to baseline, and then on, it comes back on again. So we can use the blood flow as measured by MRI to infer which areas were more active. Functional magnetic resonance imaging is useful because of its good spatial resolution. You can create a 3D map of where blood flow was happening, as opposed to EEG, where we see this smeared representation like the ceiling of the hockey arena. But because these changes in blood flow can take as long as 6 to 10 seconds, here I think the textbook makes a mistake and says a third of a second, but it's actually as long as 6 to 10 seconds. The temporal resolution of fMRI isn't as precise as EEG or ERPs, which measure things at the speed of the electrical field moving, the speed of light. But fMRI does allow us to overlay functional information onto the anatomical information, so we can identify precisely which regions of the brain had increased blood flow. One other thing functional MRI is used for is resting state analysis. So if we just stick someone in a scanner and have them not think about anything, we can use the data to identify correlations between different parts of their brain. The connectivity, the functional connectivity, if one area is more active, the other area will always be more active and vice versa. We can use this to identify networks of brain activity that work together at rest and try to understand what those networks' role might be in our cognition. So here's an example of a resting state scan that then can be used to identify correlations between brain regions and identify networks of brain areas. Another functional imaging technology that I use in my lab is called optical tomography, or functional near-infrared spectroscopy is one example of it. Functional near-infrared spectroscopy uses light, the near-infrared light, to identify the chemical makeup spectroscopy as it relates to the functioning of the brain. So we can identify properties of an object by understanding the properties of the light that gets transmitted through that object. That's what spectroscopy is. So we shine infrared light into the tissue and we measure the infrared light's properties as it comes out to determine things about the how much oxygen is in the blood. And therefore we can estimate things. Oh, and we can do this because blood that is full of oxygen and blood that is not full of oxygen differentially absorb light, different colors of light. So we can shine a couple colors of light into the brain, see how much they get absorbed, and estimate how much blood was in that area. Here's an example of a F-near system on a child, where it's a very useful technology because of its ease of use and lightweight design. So we put light in through these light injectors, a weird word. It's just a light source. Any light, like a laser pointer, can shine into your tissue. And let me try to see if I have a laser pointer right here. Yeah, so any laser pointer can shine through our tissue, and because our bodies generally doesn't absorb this red light, and so we can shine these kind of laser pointers into our brain and recreate an F-near system if we could measure the light coming out with certain detectors. And so some of the light that comes out is measured at these detectors, and the light that gets to the detector is most likely to have gone through this banana-shaped pathway through the brain. So then we can assign any data we get at that detector to that particular area of the brain. I work for a company called Blueberry that's developing the world's smallest F-NIRS device. Here's a prototype that we're working on, and here's an image of a user wearing one. So this has 3 different light sources and 2 different detectors that can measure 2 different pathways through the brain, and we can measure continuously people's brain activity throughout the day. We think this is a very promising technology, just like the portable EEG technology is. And what also is really exciting for me is when I was an undergraduate in your position, there were no companies making technologies like this. The cost was too prohibitive still, and you couldn't imagine possible careers in these fields. But now you can imagine going on to work at some of these companies or using the technologies of some of these companies in some of your own ideas or innovations. Final type of functional brain imaging we're going to talk about is called PET scanning, or positron emission tomography. This is an older imaging technique compared to fMRI that has to use a tracer injected into the blood of the individual being measured. And it attaches this tracer to certain substances like oxygen or glucose to understand how much of those substances are in the brain at any given time. So it can analyze the metabolic activity of neurons in this way. And again, we have to inject radioactive molecules that quickly decay, but have to be injected into the bloodstream before patients can do this technology. So here's a PET scanner being shown. So we inject a small amount of radioactive labeled water into the subject, and brain areas that use more blood are therefore, because water comes in the blood, going to have more of those labels. So the same idea, increased blood flow we can measure with this technology. We attribute that increased blood flow to increased neural activity. And what happens is this radioactive substance in the brain creates positrons as it decays, and these positrons shoot out in opposite directions as they're created. And because they shoot out in opposite directions, the time at which they take to arrive at detectors on opposite sides of the head can be used to infer their 3-dimensional location in the brain from which they originated. And the system can combine all these detectors' images together to recreate a map of the blood flow in the brain in this way. Not only can you measure blood flow, but you can measure other metabolites by tagging other objects besides water. So it can measure things like receptor density, metabolic activities like learning and brain poisoning, and it's widely used in cognitive function study, but not as much here at the University of Alberta. We don't have one. I think that the authors of this textbook, they have a PET scanner at Lethbridge, and so they are overemphasizing its utility in this textbook. Most of the research that I read uses fMRI these days. However, both PET and fMRI can be, and optical imaging need to particular types of analysis strategies in order to understand the data. And one really important strategy is called subtractive imaging, in which a control condition, like you're not doing anything, your brain activity is subtracted from the experimental condition, like a light is flickering. Right, so you're looking at nothing versus, or sorry, this is a light is flickering versus you're looking at nothing. Those images both show how much brain activity is present, even when we're at rest looking at nothing. This is how much blood is flowing to different brain regions. But the difference between them, they can act as their own reference, and the difference between them is most important because that shows where there was more blood flow when you are getting visual stimulation compared to sitting in the same place doing exactly the same thing except not having the stimulation. This can then be done on multiple individuals, and their brains can be aligned to average over multiple individuals to create maps of brain activity that you might have seen in popular media or in research papers. Then we can infer in this task compared to the control condition where there was more blood flow. Next, we'll talk about chemical and genetic measurement of brain and behavior briefly. So as we know, neurons' activity is regulated by their genes because genes encode all these proteins to be synthesized within the cells, and then different receptors on the outside of the cells, different shaped cells. So genes can control the cell's productions of chemicals as well, like the amount of dopamine they might produce. So we can possibly relate behavior to the genes that create these changes and to the chemicals inside and outside of the cell. One technology animal researchers use and some humans to study brain chemistry is called microdialysis. What we want is to pull extracellular fluid out of the brain to understand the chemical constituents of it. And so we can either pull out some cerebrospinal fluid and then study it, or we can use this technique to understand the cerebrospinal fluid online in a task. So a semi-permeable membrane is placed in the brain, and fluid, salt water kind of fluid that resembles cerebrospinal fluid, passes along the membrane. And because there's more concentration of chemicals in the extracellular fluid inside the brain than on the inside of this membrane, the extracellular molecules that are of greater quantity diffuse across that membrane into the salty fluid that you're passing through, and then you suck that salty fluid back up into your system and measure the chemicals in it. And then you analyze the number of chemicals at any given time to estimate how much of that substance was in the extracellular fluid. So here's the fluid coming in. It's going through this system before it comes out, and there's the semi-impermeable membrane here. And because there's higher concentration of these ions outside of the semi-permeable membrane, they're able to traverse it inside, and they enter this fluid and go up into the analysis machine. So this can be used to study, for instance, like the amount of glucose in the brain at any given time. A related technology is called cerebral photometry, in which instead of this dialysis machine with a semi-permeable membrane, a small electrode, a pair of electrodes is passed, a carbon fiber and a metal electrode, and current is passed through them. And because different chemicals in the surrounding tissue will lead to changes in the way that that current passes from the carbon to the metal electrode, the particular substances used in those electrodes can therefore be used to identify specific neurotransmitters and their quantity online in a task. So by measuring the electrical signal coming off of these electrodes that are specifically designed to, say, measure dopamine, one can estimate the ongoing amount of dopamine. Finally, we can measure gene sequences, so variations in particular genes and how they relate to brain behavior. So we could study twins and adopted children and allow us to tease apart environmental and genetic contributions. We can also study the alleles of genes and see who has particular alleles and how that relates to their behavior. For instance, there's genes for BDNF, brain-derived neurotrophic factor, an important aspect of our neuroplasticity. And there's two alleles, the 66 MET and the 66 VAL, that seem to differentially affect our production of BDNF and lead to differences in learning. And some other colleagues showed that the MET allele is associated with reduction of hippocampal volume in humans and poorer memory for events. So when we think about people that might have poor memory or poor attention or poor cognition in some domain, we might start thinking about how this might be related to the production of some certain protein by a particular genetic change. And we also know that the expression of genes can be changed by environment or experience, and these can even be passed on to our progeny. Things like chronic stress or traumatic events or disease can have cumulative experience effects on how our genes work and produce. So twins, when they are born, they have nearly identical gene expression, but over 50 years of age, they start to have very different gene expression because of different life experiences. And other colleagues have shown that the maternal attention given to rat pups alters their gene expression in adults, leading to differential hippocampal functioning. Finally, we're going to compare these techniques together, this cacophony of different techniques, and why researchers might use one or the other in different places. If you go on to take part in some research and learn how to do research in one of the labs at the university in neuroscience, you'll likely gain expertise in one or more of these techniques. And to really gain expertise in them, you really have to have that hands-on experience, which is why we want to push to have more research labs, more labs in our courses in psychology, like my 375 course, where you can do EEG. So why might we use one technology or another? Well, some researchers are focused on the structure and some are more focused on the function. And some might be interested in electrical activity and how it relates to behavior. It depends on what your research question is. You also might want to consider how much temporal resolution you need, or how quickly you need to measure what you care about, how much spatial resolution you need for your question. If your question is about where something occurs, then you do need good spatial resolution. And you care about the degree of invasiveness of the technology, depending on whether you'll be able to do the research or not. And perturbing the brain is usually less costly than acquiring one of these large imaging technologies, because they require very expensive equipment. Like the MRI machine costs millions of dollars. We use animal models to study disease in some cases. So some disorders are very easy to model, like stroke, in which you can inject blood into the brain of an animal. Behavioral disorders like ADHD are more difficult to recreate in animals. And ADHD is a developmental disorder characterized by impulsivity, hyperactivity, and inattention. And there's been a rat line that's created called the SHR rat, which is a good model for ADHD. They have abnormalities in prefrontal dopamine innervation, and these correlate with hyperactivity in the rats. And researchers can use drugs like Ritalin, methylphenidate, to reverse these abnormalities, both in children with ADHD and in these rats, showing this really useful way to use animal models to study behavioral disorder. That brings us to the discussion of why and when we're going to use animals for research and how we ethically govern our animal research. Most governments on Earth regulate the use of animals in research in their country. And universities and other research organizations usually have additional rules related to animal use. So two important issues related to animal research are, do animals get the same problems as we do, or do the models that we recreate to replicate these problems actually show what's happening in our own brains? And then also, how ethical is it to use animals for these kinds of research questions? The Canadian Council for Animal Care has 4 guidelines for when to use animals for research. So for instance, we need to only use animals for research, teaching, and testing if it contributes if it contributes to environmental knowledge of environmental principles or biological principles or development knowledge, it can be reasonably expected to benefit humans, animals, or the environment. So there needs to be a benefit to doing animal research, not just because you're curious. It has to further our understanding of the environmental, biological, or human understanding. And the better we care for our animals and the more standards we have for their health and wellness, the more credible and reproducible our research will be. And acceptance of animal research by the community depends on maintaining confidence in the process of justifying animal research. So we have to, that's why I'm telling you, for instance, right now, the different types of governing bodies that govern our use of animals in our research, so we can better understand the checks and balances that we have. And we only use animals if we can't do it any other way. And we use the most humane methods possible, the smallest number of animals possible to obtain the information that we need. So in the United States, there is a governing organization called the a governing piece of law called the Animal Welfare Act. And the NIH administers the Health Research Extension Act. Any North American university that receives government grant support needs to provide treatment for vertebrate animals and methods to show that it's ethical. However, even with all these institutional checks and balances on our animal research, there's still a considerable controversy about our use of animals for research. And this debate centers on issues about morals and biology and law. And it's important for you to understand, because in this course and in the other courses you'll take in psychology, you'll come across a lot of animal research. You might volunteer in some labs on campus that do research in rats or birds or other animals. And you want to understand what the benefit is of using these animals for research and how we're mitigating the cost to the animals. Okay, thanks very much for listening to this tour through the various research methods that we use in neuroscience. Have a good rest of your day.